Hello, and thank you for joining us for today's webinar hosted by ISEH. My name is Heather O'Leary, and I'm an assistant professor at Indiana University School of Medicine in the Division of Hematology and Oncology, and my lab studies the microenvironmental regulation of normal and malignant hematopoietic stem cells. I will be your moderator for today's session entitled Dive Deep into the Niche, which is a follow-up session from our 2018 technology session. Our presenters today are going to share their technical and intellectual expertise and useful tips for performing state-of-the-art imaging of the hematopoietic niche. If you have any questions at any time during the presentation, please feel free to type your questions into the question or chat box, and we are happy to answer them during the presentation, and there'll be time after the webinar to answer these questions as well. So on that note, it is my privilege to introduce our first speaker today. So our first speaker is Sandra Pinho, is a research associate working with Dr. Paul Fernet at the Albert Einstein School of Medicine in New York. Prior to joining Dr. Fernet's lab, she obtained her PhD in epigenetics and stem cell biology from the Imperial College of London. During her postdoctoral training, she was awarded a Druckenmiller Fellowship from the New York Stem Cell Foundation, which allowed her to investigate the identity and roles of factors in cells that regulate hematopoietic stem cells and constitute the hematopoietic niche. During these studies, she acquired extensive training and techniques relevant for imaging the bone marrow, resulted in several publications, and we look forward to the insights that she'll share with us today.
Thank you so much for the introduction, Heather, and thank you, ISEH, for the introduction. So since I'm the first panelist, I'll very briefly introduce the concept of hematopoietic stem cell niche, and later on I'll let you know a little bit about the 3D imaging technology that we've been developing and optimizing in Paul's lab to successfully image hematopoietic stem cells in their native microenvironment. So, as you can see illustrated in this slide, in homeostasis, hematopoietic stem cells basically reside inside of the bone marrow, where their behavior is controlled directly or indirectly via regulatory signals in the form of bound or secreted molecules that basically emanate from the surrounding niche cells. So this is a very active research field nowadays, and several niche regulatory cells and factors have been identified. Emerging evidence highlights the role of the vasculature, but also periarterial and perisinusoidal stroma cells, but also the role of the nerves, Schwann cells, adipocytes, as key regulators of hematopoietic stem cells. So, but also the actual hematopoietic progeny themselves has also been shown to regulate hematopoietic stem cells' behavior, such as macrophages, neutrophils, regulatory T cells, or megakaryocytes, as some of the studies from Paul's lab have been showing. So later on during the talk, I'll let you know a little bit about some of these niche studies that basically rely on powerful imaging techniques and computational analysis. But one of the problems we've been having, one of the biggest limitations in the study of hematopoietic stem cells in their native microenvironment is basically the optical opacity and the calcified nature of mammalian bones. So in contrast to studies in animals like C. elegans or zebrafish, as you have here, C. elegans, that feature intrinsic optic transparency, the nature of the mammalian bones has basically limited us to image snapshots of the cell organization of thin sections. So this really does not allow us to have access to 3D information of tissues within mammalian bones. So recent methodologies that have employed the use of high-resolution 3D imaging of whole mount tissue or employing intravital microscopy, tissue clearing techniques have allowed us to acquire the same type of optical resolution as, for example, in zebrafish or C. elegans. So another limitation we've been having in the field is that up to very recently, we didn't really have exclusive markers that allow the imaging and tracking of endogenous HSCs, but also the niche cells. So here you have some examples of key recent studies using reporter mice to label adult bone marrow hematopoietic stem cells. And out of these 13 studies, already 8 of these reporters were already tested by different types of imaging, such as clearing or whole mount immunostaining coupled with confocal techniques. So In Paul's lab, in order to gain detailed insight into 3D structure of the hematopoietic stem cell niche, we've developed a whole mount immunofluorescence imaging technique of the actual sternal bone marrow. So here you have a longitudinal view of a mouse femur and a sternum, illustrating the arrangement of blood vessels and nerves within the bone marrow cavity. So unlike the long bones, which are mainly occupied by adipocytes in adult humans, the sternum exhibits a rich hematopoietic activity in both the mouse and the human species, which makes it actually a very useful organ and site to study hematopoiesis. So since this is a practical session, I'll take you very briefly a little bit through our protocols that basically starts with the careful collection of the tissue, as you can see here in figure 1. Basically, after carefully collecting the sternum, you remove the tissue from all the fat and muscles, and you get rid of the ribs by cutting along the costal cartilage, and you very carefully expose the bone marrow of each segment by cutting it along the sagittal plane, as you can see here in red. With the help of a blade and endoscope. So this is actually a very simple, it's a delicate process, but it's very simple if you compare with the rest of the processing for example, for long bones. Also, in terms of the actual procedure, it's a relatively short procedure. In 4 days, you can be ready to go to the microscope. So after the collection and fixation, a very short fixation of the tissue, you go through standard blocking permeabilization, and you do your immunostaining with primary antibodies. And then if you require, you might have to do a secondary staining, for example, with streptavidin. And in day 4, you are ready to go and image. You can do the entire procedure using 96 well plates, so that's pretty practical. And the imaging of the sternum, actually the whole mount, has several advantages compared with other methods because it doesn't really require harsh decalcification steps. As you can see in the diagram, these allow us to preserve the surface antigens and also endogenous proteins such as GFP and cherry, and we can get imaging up to 200 microns deep by confocal microscopy. So in terms of practical tips that we can share that we think are important is if you're interested in imaging the vasculature, inject IV or antibodies 10 minutes at least before tissue collection. It sounds a little bit basic, but always spin your primary antibodies to remove fluorochrome aggregates if you don't want to end up with a mess. Whenever possible, use a new and freshly opened vial of the CD150 antibody. This seems to be one of the most sensitive. And in our setup with the sternum, it really helps if you use upright microscopes with immersion objectives. So as I mentioned, using the sternum imaging, we can very easily look to the bone marrow vasculature. Here, for example, you have imaging of the sternal bone compartment where you have the entire vasculature labeled by CD31 and CD144 in red, and in green you can see arterioles labeled by Sky1 bright expression. This technique helped us in the lab to show distant function for vascular structures of the bone marrow. A previous postdoc in the lab, Yuya Kunizaki, showed that arterioles and perivascular mesenchymal stem cells play an important role in maintaining hematopoietic stem cell quiescence. So this slide shows a representative imaging of a whole mount bone marrow staining with the vasculature in blue, and arterioles are in green, marked by nesting GFP bright, and hematopoietic stem cells can be identified by the expression of CD150 here in red and the absence of expression of CD48 and CD41 and hematopoietic lineage markers here in pale yellow. So we can also combine our imaging with computational simulations of randomly distributed hematopoietic stem cells. To do so, we use images of our sternal bone marrow of interest. You have one type of niche that you highlight. Here we have megakaryocytes in red. And you can establish a random model by first defining a binary spatial map of the initial structures from these images of bone marrow segments. So to define the random distribution, basically in which the HSCs are not preferentially localized near any of the marrow structures, we randomly place hematopoietic stem cells within the unoccupied regions of the spatial maps and measured the Euclidean distance of each HSC to the near niche structure, in this case megakaryocytes. So using this type of analysis, we are also able to observe that hematopoietic stem cells seems to be frequently localized near megakaryocytes in the bone marrow. And you can see here by the distribution, we observed that approximately 30% of hematopoietic stem cells are in close proximity with megakaryocytes. And this is actually not random, as by a random computational model, we would only expect around 11% of hematopoietic stem cells. And this is actually extremely statistically significant if we use appropriate statistic methods of distribution like the Kolmogorov-Smirnov method. So also, as I just mentioned, the field is actually very dynamic, and more niche structures keep being identified. So we can also use this method to analyze distribution of HSCs not to one niche structure, but more than one. In this case, we can generate 2D probability graphs of distribution, for example, of HSCs relative to megakaryocytes, but also to arterials. And these allowed us to, for example, identify that in the bone marrow, hematopoietic stem cells in steady state are mainly in two configurations, where approximately 15% of them are within 25 microns distance of megakaryocytes and arterials. Basically, it's a niche compromise of both structures. But also, and this is actually very different from a random computational generated model, as you can see here on the right. And in the second configuration, we saw that approximately 21% of the hematopoietic stem cells are localized in close proximity with megakaryocytes, but further away from arterials. So it suggests a niche that is spatially distant from the arterial niche. And most recently, we used this imaging analysis to investigate the regulation of distant subsets of hematopoietic stem cells by bone marrow niches. So we use von Willebrand factor VWF EGFP mice, which was where VWF was shown to mark a subset of hematopoietic stem cells that exhibit platelet and myeloid bias. And as you can appreciate in this image, VWF labels hematopoietic stem cells, but also a subset of hematopoietic stem cells, but also megakaryocytes. And not going into details about this study, but it was very interesting to see that VWF positive myeloid bias and VWF negative hematopoietic stem cells are located in and regulated by separate bone niches in the bone marrow, occupied by megakaryocytes or by arterials. So finally, I would just like to acknowledge everyone in Pulse Lab, all the present, and also some of the former members that helped to really establish and optimize this external bone marrow imaging. So thank you, and thank you all for listening.
Thank you so much, Sandra. That was great. So our next speaker is going to be Mehmet Sacma, who's focusing on stem cell research in the Institute of Molecular Medicine, Stem Cells and Aging at Ulm University, headed by Dr. Hartmut Geiger. And currently he's working on projects examining hematopoietic stem cell biology and aging of the niche. And furthermore, he develops methodology for simultaneously imaging of HSCs, progenitor, and various niche components in bone from both large field to cellular levels. He holds a diploma in molecular biology from Ulm and a scholarship from the federally funded Avencia Scholarship Foundation in Germany. So thank you very much.
Thank you, Heather, and hello to everybody. I want also to thank the organizers that they give me the possibility to present our methodology entitled Imaging of HSCs, Progenitors, and Multiple Niche Components on Cellular Level. When we have a look into the bone marrow with a simple Doppler staining, we see a lot of different cells. Some are bigger than others or are elongated in their nuclei. But we cannot concretely say something about the types of the cells or even show which one is a very rare stem cell. That's why we need proper techniques to identify specific cells in the bone marrow. For example, when we add endothelial markers here in blue, we see that these elongated cells appear as endothelial cells. And when we add the hematopoietic marker CD45, we see that a lot of cells in bone marrow have hematopoietic origin, but not all like this. Because these are stromal cells, exactly, they are leptin receptor positive cells, which are known to be regulating HSCs. And with this image, I want to show you our technique how to do this. We also use and further developed whole mount immunofluorescent techniques in a modified version. First, bones were harvested after perfusion with PFA and were post-fixed for 24 hours. Subsequently, bones were embedded and were snap frozen in liquid nitrogen and can be stored at -80 longer time. Then the bones were shaved on a cryostat until the bone marrow cavity was fully exposed. Afterwards, the staining procedure started. We are using 6 to maximum 20 volt plates for washing steps to ensure that the probes are always in liquid. For incubation, we use always PCR tubes or mini EPIs, and depending on the used antibodies, we have different incubation times. And finally, the probes can be imaged by confocal or two-photon microscopy. The labeled hole mounts were placed cut face down onto a 4-well microslide and were covered in antifate to prevent tissue desiccation. After the imaging, the data can be analyzed by image analysis software like IMARIS, Velocity, ImageJ, or others. The technique is quite fast and reproducible. Since femurs and tibia are commonly used for FACS experiments, with this approach we can at the same time use the sterna and humeri for histological experiments and can achieve the first images after 4 days for simple stainings. We don't need decalcification and clearing. Finally, as you will see in my examples, the architecture and morphology of the entire bone and bone marrow is highly preserved. With our method, we can currently image with a conventional confocal laser microscope 7 colors simultaneously. We could do this for DAPI, GFP, YFP, PE or size 3, red and far red and interference contrast. And theoretically, with an additional detector, we should image markers additionally in more than 750 nanometers without an extra plugin or other further softwares. Now I want you to show some examples, and let's start with the imaging of endogenous HSCs. We are also using the same antibody code which Sandra introduced, and here we see a sector from the bone marrow, and on the right we see the endosteum, which is the border to the bone matrix. In blue we see the vasculature, and the arrow is showing a slam-marked HSC close to the bone surface. The antibody code on the top shows that the HSC is negative for the white exclusion markers, in total here with 8 antibody listed. So we can find HSCs by this antibody code in our histological images. On the right, in gray, we see the bone. In blue is the vasculature here. The big red cells are megakaryocytes, which are also positive for the stem marker CD150. We can mark the HSCs, as you can see here, with the yellow dots. So when we marked our HSCs based on our antibody code in the bone marrow, the software can recognize the 3D information of each stem cell. And can, for example, easily show and measure the nearest distance to the endosteum on the right. So it is possible to quantify relative distances of HSCs to multiple niche components. And here, for example, we measured in this way the distances of HSCs to the endosteum and see that they are more distant in the old bone marrow compared to young wild-type mice. The next chapter is about the possibilities of imaging hematopoietic progenitors with this technique. We established an IF staining for early lymphoid progenitors with IL-7R alpha and lineage antibody combination. In whole mounts, as you can see here, of young and old long bones, we can detect the localization of lymphoid progenitors in the bone marrow, which are positive for IL-7R alpha and negative for the lineage antibodies. With the HSC staining together, we can decipher the spatial relationship of early progenitors and HSCs in the bone marrow. Since vascular niches are important in regulating and maintaining HSCs, it is important to image blood vessels in the bone. This can be done with different markers like PCAM1, polycadrine, endomycin, laminin, or FAP-B4. And with the software, for example, we can measure the volume of the vasculature based on the signal in the 3D reconstructed image on the right. For example, in the diaphysis here, we see no differences between young and aged bone marrow vasculature volume. Another application, we see age-related alterations of vasculature in the epiphysis, and we can mark the vessels, and the software can measure their length, diameter, or orientation to the bone axis. With the HSC staining on top, we can measure the relative distances of HSCs to different blood vessel types, like to arterioles here on the top or to sinusoids here below. Another important niche component are perivascular cells. In a modified version of the method, it is also possible to do two-photon microscopy of long bones, in which we can image in nesting GFP mice, for example, perivascular cells and the bone matrix together much deeper into the tissue, and do intravital imaging as Myriam will introduce later with her method. Here we see HSCs close to green nesting GFP high cells, which are around arterioles, also Sandra showed before. Signaling molecules can show potential interaction between bone marrow cells. Here we see a sector from the trabecular part of the bone. We might see mainly osteoblasts and osteocytes. And the extracellular matrix protein osteopontin is, for example, mainly found in this region of the bone marrow and is mostly produced by bone lining cells, like seen here on cellular level in the magnified left image. And also not signaling proteins, like for example the ligand JAK2 here in red, which expression is lower in H periarteriolar cells in the lower images. Another application for this method is the imaging of fluorescence proteins without restaining them. We can image, for example, the very dim GFP signal of nesting GFP low cells and show that they are mainly leptin receptor positive and are around here blue sinusoids. Also, label retaining cells, based on their ability to retain the H2B GFP signal label in vivo, can be visualized without restaining the GFP molecule. Here we see a very rare old label retaining HSC. It is also possible to visualize transplanted HSCs. In this experiment, we sorted RFP positive HSCs from young and old RFP mice and transplanted them in recipients. After 6 weeks, we see that the engraftment of young donors is better, especially in the endosteal area, because we see here more RFP positive progeny in the recipient bone marrow, here in the middle images. On the right, we see again the vasculature. We can also image simultaneously competitive transplanted young CFP positive here in cyan and old YFP positive HSCs here in yellow in different recipients, like on the left in young and on the right with old recipient mice. And further applications with the method, it is also possible to show the cell cycle status of bone marrow cells in different regions with the proliferation marker Ki67, for example. We see here, for example, the HSCs with the proliferating marker Ki67, like I said, in 5FU conditioned bone marrow, where we can see also very enlarged vasculature in blue. And with this, I want to come to the end of my presentation and thank to all the people who contributed to this work, and thank you for your attention.
Thank you so much for all of that information. So I'm excited to introduce our third speaker today, who is Konstantinos Kokkaliaris. So he studied biology at the National University of Athens before moving to the University of Edinburgh for his master's degree in life sciences. He received his PhD from the Ludwig Maximilian University of Munich under the supervision of Tim Schroeder, with whom he continued working as a postdoctoral fellow at the Swiss Federal Institute of Technology. His main interests are the complexity of the bone marrow microenvironment that support hematopoietic cells throughout life, and during his postdoctoral work, he characterized the spatial distribution and marker co-expression of bone marrow niche components using novel quantitative multicolor full bone marrow imaging. At last year's ISEH meeting, he was awarded with the Eugene Conkright New Investigator Award, and he recently joined David Scadden's lab at Harvard as a postdoctoral fellow.
Thank you, Heather, for the very kind introduction. So, as Heather said, I am currently a postdoc at David Scadden's lab at Massachusetts General Hospital and Harvard Stem Cell Institute. But today, I would like to talk to you about some of my previous work during my first postdoc at Tim Schroeder's lab at the ETH Zurich. So as nicely highlighted by Mehmet and Sandra before, bone marrow imaging is a very challenging technique. Classical approaches include simple stainings in plastic or paraffin-embedded sections. And during the recent years, immunofluorescence of frozen or decalcified sections is another method that has lately progressed significantly and yields very good results. However, if we want to perform imaging studies and bone marrow imaging, it is very important to consider and try to address several critical parameters, which often are limiting factors for currently available technologies. One of these is, for example, the tissue morphology preservation, which in some methods can be problematic, and if we want to study the distribution of a specific cell type in the bone marrow, we have to make sure that our morphology preservation is very good. Another important consideration is the section thickness and the imaging depth, because if we have 10 micron thick sections, for example, 10 microns is the diameter of an HSC, so we don't know in 3D what is above or below, so we might miss important information. Another important aspect is try to decrease autofluorescence as much as possible. This is specifically important for UV and 488 excitable fluorochromes, while at the same time try to increase the number of acquired markers, because this would give us higher multiplexity. It is also very important to have full bone information or full organ information, just to be able to detect potentially different behavior or differences between anatomical locations while combining this with quantitative methods. So my reason for diving into this, to be in line with today's title of the webinar, was my interest for studying the bone marrow hematopoietic stem cell niche, which until today, as Sandra said, is a quite controversial topic. So to study HSC localization in relation to different putative niche markers, we need a method that ticks all these boxes. So preserves tissue morphology, has deep tissue imaging, autofluorescence, high number of markers, full bone information, and quantitative analysis. So such technique did not exist 6 years ago, so I joined forces with another postdoc in Tim's lab, Daniel, who had a strong background in bone biology and started already developing methods for this. And this joint effort resulted in establishing an approach that allows us to always image full bones, as you can see here on the left, while staining for different types of epitopes like membrane, ECM, even cytokines, and we can also stain nuclear epitopes. We can also preserve endogenous proteins or stain for them, and we can go down to microstructures like cilia. So we basically have subcellular resolution for our imaging, and depending on our excitation laser, location in the bone, and fluorochrome used, we can go several hundred microns deep inside the tissue using optical clearing. Our approach is not limited to bone. Actually, bone is the most difficult tissue to manipulate and section, but we can also apply it in brain, muscles, lung, or and virtually any other tissue. One important aspect of this approach was trying to multiplex as many fluorochromes as possible, and for this it's very important to select fluorochromes that robustly work with epitopes of interest, and also take special care for the clearing agent and mounting media to be compatible with what we are using as fluorochromes. Because, for example, it is known that specific clearing agents are only compatible with specific fluorochromes. So here we have extensively tested different classes of fluorochromes and also different mounting and clearing media, and we managed to have an approach that basically works with all while preserving endogenous proteins, and this is by using clearing TBE, which is 2.2 thiodieethanol, and this allows us then to do multicolor imaging, and as you can see in this slide, we can do 7-8 color imaging by, for example, marking for collagen, which stains bone, DAPI for nucleated cells, GFAP for non-myelinated Schwann cells, we can have osteocalcin for osteoblasts, SCA for arteries, arterioles, and hematopoietic cells, KIT for hematopoietic cells, and 105 for sinusoids and other vessels. We also have IL-1 receptor 1 in this staining, but doesn't project very well, so I didn't include it here. So basically 7-8 color stainings are feasible, are very time consuming to set up, but if you have sequential immunostaining rounds during staining and blocking antibodies of the same species and having sequential acquisition rounds in the microscope, this is basically feasible. This is very similar to what Mehmet showed before, and also in our case this is a conventional SP8 like a microscope which is basically available in most facilities, and nothing basically super fancy for acquisition. Our approach is a bit more time consuming, it's more lengthy, so we isolate the bones, we fix overnight, we decalcify for 2 weeks, and then we do the embedding and the sectioning for full sections, and then the immunostaining, depending on the complexity of the markers that you want to stain, takes up to 8 days. Clearing for 2 or 3 days, and then the imaging also takes time, and this is without taking into consideration the computational analysis. So after the image acquisition, what happens is that we do the post-acquisition image analysis, and in this case it is segmentation and distance quantification. So I show you here an example where we have stained for KSL cells, these are enriched in hematopoietic stem and progenitor cells, and they are expressing c-KIT, SCA, but they don't have lineage. So what happens as a first step, we are segmenting on cells of interest. In this case, we have c-KIT, which is the easiest marker to segment, and using IMARIS, we can extract all the necessary features, and then we can plug them into our own software, which was programmed by Leo, who later joined our team as well. And the software is called Excite, and then you can quickly process the data and identify relevant features. And from this, we can identify which cells are actually expressing KIT and at the same time SCA, but have absence of lineage markers. As the next step, we can segment on a surface of interest. In this case, we have collagen with marked bone, and we can create channels that basically gives us the distance from all individual objects. To visualize what I just said, we have a full bone staining of KSLs. Here we have a magenta object, which is a KIT positive, SCA positive cell here, and also doesn't have lineage. And we can identify hundreds of these cells and thousands over the bone, and then we can at the same time segment on GFAP non-linear Schwann cells and measure the distances of all these cells from this structure and also from collagen 1, which marks bone. So having this technology up and running, we took a step forwards and we wanted to dissect the bone marrow niche, and for this we tested almost 300 different primary antibodies. Some of them worked, we were lucky, and we used some to mark osteoblasts or bone cells, chondrocytes, endothelial cells and perivascular cells, neuronal structures, stroma cells, and matrix. So for all these markers, we have full bone information. We know exactly where the populations are located throughout the bones in different anatomical locations, and we also have quantitative data for their co-expression with other markers. And this basically allowed us to clarify some of the confusion in the field. As an example here, I show you a panel from the endothelial markers, and some of them have been used interchangeably in many labs as panendothelial markers. So if we stain for CD31, we see that indeed it stains for arterioles, also megakaryocytes and other cell types, but also for the long diaphyseal arteries, and it's also present in sinusoids and the central sinus, although the signal intensity is a bit weaker. On the other hand, CD105 is also a marker that has been used as a panendothelial marker, but as you can see here, although it stains some of the arterioles, it doesn't stain all of them, as marked here by these arrowheads. It's absent from the long diaphyseal arteries, but it's very strong in sinusoids and the central sinus. SCA1 is a typical arterial marker, and indeed is expressed on all arterioles or most arterioles in the long diaphyseal arteries. And we have also another marker, which is SM22, which marks the smooth muscle cells that are wrapped around the arteries. So this is exactly where we see it. So as I said before, we have, for the different markers that we have tested, we have these quantitative maps that allow readers, if they're interested in specific cell type, like capillaries, to know exactly which cell type, which marker they can use. And the most interesting part for me starts now after having done all this, which is again the identification of the HSC localization in relation to different markers. But due to the lack of time, I will just stop here. And I would like to thank Tim Schroeder for being a great mentor and for creating a very supportive niche for playing with different technologies in the lab. And of course, Daniel and Leo for being fantastic colleagues during this long journey and for sharing the hard work, long weekends, troubleshooting, and because without them, this studies would not be possible. I would also like to thank all members of the Schroeder Lab for their intellectual contribution and for their support, and thank you for your attention.
Thank you. That was really interesting, Konstantinos. Okay, so our final speaker today is going to be Myriam Haltali, and she's a PhD student in Christina Losalso's research group at Imperial College of London. Her research is focused on investigating the role of the bone marrow microenvironment in response to hematopoietic stem cell malaria infection. And she uses a range of techniques, including intravital microscopy of the mouse calvarium, to address how the proliferation dynamics of HSCs change in response to infection, how this alters their long-term function, and what role the HSC niche plays in this. These projects could have important implications for those who survive severe infections, and when Myriam is away from the lab, she apparently loves to travel, and more recently has started to get into baking. So we're excited to see all the interesting science you have to tell us about today.
Great, thank you so much, Heather, and thank you for giving me the opportunity to contribute to the panel today. So in my short presentation, I'm hoping to touch on some of the aspects of intravital imaging that we specifically do in our lab. I am aware, and it is important to note, that there may be differences in the technique from lab to lab, but hopefully by going through our typical imaging setup, experimental design and acquisition for different circumstances, and the image analysis techniques that we tend to use, I hope I'll be able to highlight some things that our research group has found really useful and some tips or tricks that you might find useful if you're considering to use this technique. So over the past few years, intravital microscopy of the bone marrow cellular environment has improved massively thanks to the development of various dyes, fluorophores, fluorochromes, model organisms, as well as the advance of microscopy techniques. In particular, the combination of confocal microscopy with two-photon microscopy for intravital imaging has really given us the ability to visualize thick tissues, including the bone marrow cavity, in the skull of mice, and this has really allowed us to detail the dynamics and complexities of various biological processes in vivo and in real time. So as an example here, intravital microscopy really played a huge role in these two major studies that came out of our research group over the past few years. This tissue has a main advantage in that the bone plate is very thin and does not need to be shaved down, therefore allowing non-invasive imaging of the bone marrow microenvironment without altering the tissue's architecture. So as you can see in this diagram, by attaching a specific headpiece to the mouse that is designed to fit into our imaging setup, we're able to define the imaging window, and by taking a series of Z-stacks across the calvarium, and later on stitching them together during image processing, we can create this overview of the bone marrow space. So this is, just to guide you, this is where the mouse's nose would be, close to the bifurcation. The coronal suture sits towards the ears of the mouse, and we tend to image along the central vein in the calvarium. And what's really cool is that with the mouse on the stage, we can then go back to this image and use it as a guide to then select some positions of interest within the calvarium and take some time-lapse movies to visualize the specific interactions in real time. What has also been really useful is the development of a range of reporter mice, not limited to these examples that I have here, that have really allowed us to image different niche components and various cell types in the bone marrow. So I'll just show you our imaging setup and the room that we tend to spend so many hours in, and you can see Christina just there taking some beautiful images. And so this is our microscope, and this is where we place the mouse after having induced anesthesia in a specific hood that is at the back of the room, which is equipped with isoflurane, heat mats, and the tools that we need for surgery. We have isoflurane also hooked up to the microscope and delivered through this tube here in order to maintain the mouse under anesthesia. And we have a heat pad that rests underneath the mouse and is connected to a rectal thermometer for us to be able to monitor and maintain a steady temperature throughout the imaging session. So it's important to note that the mouse's comfort and well-being is always our priority throughout the session, and we do everything that we can to ensure that. Some more technical tips. So in our lab, we attach the headpiece to the mouse's skull using dental cement. And it's really important to have patience during this step to ensure that the headpiece is well attached and in particular that you have really cleared away enough fur and skin that the headpiece, so that the headpiece can sit directly on the bone and won't drop during your imaging session. Also to ensure that it's important to ensure that the cement has completely dried before moving the mouse to the microscope stage. Upon exposing the skull, what you will also notice is a very thin membrane of skin that sits on the bone, and it's important to clear this away before you start acquiring any images, just with PBS and a cotton bud. And you will really notice a drastic improvement in the quality of images that you can obtain. And another great advantage of this technique is that we have the ability to recover the mice and image again a few days later, for example. And if this is something that you're interested in doing, it's important to try and work as quickly and as efficiently as possible to minimize the time that the mouse spends under anesthesia. And this will just ensure that your mouse is able and fit enough to cope with further imaging sessions. So I'll now focus a bit on the planning and data acquisition. So I've written here that it's important to define and refine your specific research question in order to determine the type of imaging experiment you need to do, and the specific parameters you need to use. So this isn't a really easy technique to carry out, and it really does take a significant amount of time and effort. So ensuring that you've made a good plan means that you'll always achieve the best quality of data at the end. So I'll take you through two main types of experiments that we carry out in the lab. So firstly, these tile scan overviews of the calvarium bone marrow and the time-lapse imaging of the specific areas. So as I mentioned before, tile scans really provide us with a Google Maps overview of the bone marrow cavity. And what's great is that you're able to image a number of components in different colors by setting up and acquiring various tracks on your system. So in the example here, we have healthy cells in yellow, leukemic cells in red, and bone and the vessels in blue. One suggestion that we can give is that you want to ensure that within an experiment or project, you're trying to keep a consistent imaging window throughout. So if we look at this diagram of the exposed calvarium, what we typically do in our lab is try to image from the coronal suture at the back of the head towards the bifurcation. So it's just important to have this in mind when you're placing your headpiece to ensure that you don't cover up any areas that you will need to image. Furthermore, it's really important to ensure that the depth of your Z-stack will capture all the information that you need. So it's important to remember that the calvarium is not a flat piece of bone, so you'll need to check the Z-stack that you have set up across different areas along the calvarium to make sure that you're not losing any important information. And then in terms of processing and analysis, we have found that cropping the tile scans to remove autofluorescent signal and bone signal from other channels can really make a huge difference to the tile scan, and particularly this is important before quantification. So you can see in this maximum projection of an uncropped tile scan from a Nestin reporter mouse that we have a lot of autofluorescence coming through from the osteocytes in the bone. And if I was to quantify this image directly, it would not really give me an accurate readout of how many Nestin positive cells there are. But when we take a look at the same tile scan that has been cropped, you can see a huge difference, and we can clearly see the Nestin cells, and a quantification of this tile scan will be much more reliable. So we usually carry out this cropping manually in ImageJ, and this can be quite time consuming, but so far we haven't found a good way of automating this process. However, creating little macros for the various parts of the cropping process in ImageJ can help you to speed up the task. This is also quite a subjective process, so I might crop the tile scan in a completely different way to one of you guys, and I might also crop it differently today than I would tomorrow morning. So I would really recommend doing the processing in batches based on your experiments and trying to do it within the same sitting, just to try and keep things as consistent as possible. And in terms of quantification, in our lab we tend to calculate volumes of cells and distances, for example, using a program called Definiens or Imaris. Moving on to time-lapse imaging, again, how you set up your time-lapse will really depend on the specific aim of your experiment. You need to consider how many positions you want to acquire, and how many positions you actually can acquire will depend on how many tracks you have, the averaging, which is basically a way to reduce noise within the images. So the higher the averaging, the longer it will take to acquire the image, and how frequently you would like to take an image within your time-lapse. So this particular time interval will really depend on the type of cells or the specific event that you're trying to capture. So here I have a few examples of different experiments that we do in the lab, and I just wanted to use these to illustrate how different the time intervals can be. So in the first video, we were imaging HSCs, or hematopoietic stem cells, that have been transplanted into irradiated mice. And given the fact that they move so slowly, we were able to take one image every 3 minutes, but still be able to follow the tracks of each cell really nicely. We also do experiments where we measure the leakiness of vessels by injecting a vascular dye and imaging over a set period of time, so that we can quantify how much of that dye leaks out into the parenchyma. So in this case, we took one image every minute for a period of 10 minutes, and that was enough for us to be able to see how much of the dye was leaking out and quantify it later on. And in this last video here, we were looking at the competition between healthy bone marrow and leukemic cells, and as you can see, these cells move really fast. So in order to capture information accurately and be able to track the cells accurately, we were taking an image every 30 seconds. So it really depends on your specific research aims and what you're trying to address with the imaging. It is really important to bear in mind that we can only right now select positions for time-lapse imaging before starting acquisition. And unfortunately, this can't be changed during acquisition. So once you've selected your positions, you're committed. And this can really cause a problem if, for example, you're tracking a cell and it moves out of frame or deeper into the bone marrow while you're imaging. So it's just something to bear in mind when setting up your time-lapses. During acquisition, you will often find that your images might drift or move due to the mouse's breathing, the headpiece becoming loose, or movement of the stage during your session. And in terms of processing and analysis, one thing that our lab finds really useful for videos is registration, which is essentially the process of using structures within the image that are the most stable to essentially anchor the time-lapse and cancel out the drifting that can be seen. So for example, if we take a look at the movie on the left, you can see that it's kind of shaking and moving down. And after applying the registration, you can see on the right that we've completely stabilized the movie and it's much easier to look at and appreciate. And we just carry out this process using one of the tools on ImageJ. Other things to bear in mind is to watch out for bleaching of various fluorophores. For example, we know that tomato bone marrow tends to bleach quite quickly, so we have to limit the exposure to the laser as much as possible. And this can often be corrected post-acquisition in ImageJ with bleach correction. We mostly carry out analysis of time-lapse movies using ImageJ or Amaris. For example, we track cells, calculate distances, or quantify leakiness. And there are many, many other things that you can do. It really depends on your experiment and or research aim. So I'll stop there, and I hope that I've provided some useful insight into the more practical and technical side of intravital microscopy. So I just want to quickly thank Delphine, Chiara, and Sarah, who provided some of the images that we've seen through the talk, and I'll be happy to help with any questions.
Great. Thank you so much, Myriam, for all of that insight, and thank you to all of our presenters. Right now, if you want to start adding in questions to the chat boxes, you're welcome to do so. If not, I'll probably start with questions. So all of you kind of have the same goal, but use very different models to do it. And so I was just curious if you could each speak a little bit to why you chose the methodologies that you chose. For example, some people decalcify, some people don't. And to that end, how it's different in imaging, choosing things like fluorophores and antibody concentration compared to something like flow cytometry.
Well, I can start like at least in terms of choice of the tissue, of the organ. We use the sternum, and of course, like we cannot exclude the usage of long bones. But for example, compared with the long bones, we find the sternum has several advantages, like what I was mentioning in the humans. The sternum is an actual organ of active hematopoiesis, while the long bones in humans are full of adipocytes and are really not active sites. And also we can do imaging in very fast way. We skip all this processing of freezing and sectioning, which destroys a lot of the surface markers, and is also not ideal for some of reporters like GFP or tomato. So definitely this is an advantage. Of course, if you go to other types of methods like clearing, you can include, you can have a higher tissue depth in terms of imaging, but at the same time you have to rely on decalcification of marrow extraction. So I guess in the end it's always a balance between what is your goal, if you're really focusing on more on the AJC or in the relationship with the niche, if you need to rely on reporters.
Okay, great. Does anyone else want to add to that at all? And Konstantinos, I know you said you tested like 300 antibodies. So how did you determine which ones were most optimal and concentrations and things like that for the fluorophores versus ones you decided not to go ahead and follow up on?
Yeah, I mean, I totally agree with Sandra. First of all, it depends on what you want to do. And indeed, for some questions, having deep tissue imaging might be not that's essential. But yeah, I mean, I think we decided to go with that, and it was a very big detour for us because we thought that we want to have as much information as possible to answer your first question. As possible, and then in full bones always, and the long bones are more challenging, so that was also the motivation from our side. And yeah, I mean, for the antibodies, we started with things that basically were known to be expressing specific cell types, so it was partially confirming what was out there. And yeah, I mean, we even had experiences with different, same antibody, different lot numbers not working the same. So it's very difficult to validate antibodies, but I think if you do a different, so we tried for every epitope, we tried 3-4 different companies, and that was, I think, the critical way, the critical method of how we could verify our results. So if with 2 or 3 antibodies from different companies, different clone numbers, we saw the same thing, we were confident that that was the appropriate signal, but of course we cannot rule out that decalcification affects some of them, and this is, for example, known for 150 that doesn't work in decalcified bones. So this limitation is there. It doesn't mean that everything that doesn't work for us, it won't work in general, but with these specific methods, and we have reported all these antibodies in the study, so if anybody is interested, he can go and check which antibodies we tested and if they require right fluorochromes or not, so this information is available.
Great, thank you. So Myriam, you do the live animal imaging, so I guess a question that most people would have would be, if you're doing the trial versus the time-lapse imaging, do you have to alter the number of cells that you inject, and how do you modify stem cell numbers or numbers of cells you inject versus numbers of cells you can actually image either on the tile scan or through the time-lapse?
Yeah, it took a long time optimizing the optimal number to inject in order to see anything in the calvarium. The reality is if you inject 10,000 HSCs, you're likely to find 2 or 3 in the calvarium. So it really is a massive task of patience and being able to sort as many cells as possible to inject, to give you as many to inject into as many mice as possible and give you enough kind of chance to find that needle in the haystack. But it is really difficult just due to the nature of how, you know, cells will of course not always end up where you want them to be if you inject them via the tail vein. But yeah, that's the reality. If we inject 10,000, we expect to see 2 or 3 on a good day.
Okay. And then I think the final question I have, at least for all of you, is you use a lot of different software, everything from ImageJ to Amaris, and it seems like you kind of vary between software. How do you, or how did you determine, for someone who's starting this sort of new, what software was the best for your application? Did you try multiple softwares, or how did you determine what was going to give you the best result for your specific application?
I can start. I have to say ImageJ is a really good place to start for someone who has no previous experience. There's lots of information online and lots of blogs, so you can literally type in any question that you might have, and most times, probably 90% of the time, someone's written a small article with some instructions. So ImageJ is a really nice place to start. Amaris is also pretty easy to get used to. But it really is trial and error and thinking about what your specific question is and what it is that you want to gain from your analysis.
So yes, in our case, we also try different software. This also often depends on availability because Amaris is pretty expensive as a software, so not all facilities will have it. And yeah, I think all software have pros and cons. So for example, some things you cannot, so for big data sets and for stitched files, some of them might have difficulties. although you might gain other things like plugins and software and more modules, but I think it again depends on the question. So some other software have the possibility of training your algorithm in detecting signal. Some are better in segmentation. So it really depends. And very often, because exactly we didn't feel that the available software, even commercially available software, ticks all the boxes and fulfills all our needs. We also have to program our own software, a plugin, so basically a standalone that will supplement IMARIS. And I think if somebody wants to go deep into the quantification, he needs to go to this level and program something for his own needs, because it won't be covered by what is out there. At least this is our experience.
Great. Does anybody else want to comment? Okay. Well, on that note, thank you everyone for listening to our webinar today. And I'd like to extend a special thank you to our presenters for all of their effort and their insight today. Attendees, please feel free to complete the evaluation on today's event that you'll receive via email so we can further develop ISEH webinar programming based on your feedback and input. Thank you again, and everybody have a great day.